So I have begun the recording. There was some problem that I was facing while joining this link because apparently the link that they have given was not opening up. Okay, so let me just, yeah. So I'll share my screen and we'll begin this discussion. And as a disclaimer, I would like to, tell you guys beforehand that this session that we'll be seeing today, okay, it is a little different from how I've conducted my other practice sessions. So those in the name of practice sessions, I have try to, solve previous year questions and discuss concepts that were already discussed during that week's lectures. Along with that, what I have tried to do in my other practice sessions is that I have tried to share some knowledge that was already not there in the week's lectures, right? So some additional knowledge for you guys to understand this course, that is cell biology, better, right? So that was not just for the exams, but today what we are going to do is this is a practice session, and what I have done is that I have taken simply taken ma'am's notes, and since they were not available in the PDF format to us as well. So I have, I actually started to prepare a presentation wherein I was making fresh slides, but eventually I have come to just take snapshots from ma'am's lectures, and we are just going to discuss what has been discussed in this course. Also, another disclaimer is that I think we'll be able to cover up till week 4 in these 2 hours, right? But once I hope that you guys do receive the study material that we share with you guys. Right. So that when I'll be sharing, I'll share the study material for the entire 8 weeks. So that will also in that PDF you can it can be a summary kind of for the entire course. Also because a lot of you had doubts that or had this complaint that there was no PDF that was available. So most of the slides will be there in that, right? So having said that, we'll just begin this discussion and yeah, just a moment. So for the last time, I am Arveen Kaur. I am a PhD scholar at the Department of Biological Sciences and Bioengineering at IIT Kanpur. Along with this, I'm a Prime Minister's Research Fellow, for which I have been given this chance by NPTEL and by Professor Shikha Laloria to be the teaching assistant for this particular course, that is Cell Biology, Cellular Organization, Division and Processes. And I hope you have enjoyed this course and you have gained a lot of knowledge regarding cell biology through this course. It was a data intensive course, right? So 8 weeks might not have been enough. But despite that, I hope you have learned a lot of new things, and you will do good in your exams if you have registered, right? So we'll just begin with what is cell biology, right? So what is cell biology? Cell biology is basically understanding the unit of life in in living organisms. So what is the unit of life in living organisms? It is a single cell, right? So understanding how the inner mechanics of the cell work is kind of what is meant by cell biology. Now, when we talk about the history of cell biology, uh, I I have just kept the timeline that has been discussed with you in this course, right? So, the history of cell biology is actually very, very extensive. But if you have to talk about a brief history, then we start with Robert Hooke in 1665, who first visualized cells, right? These were dead cells. They were cells that he visualized in cork, that is a kind of wood, right? After that, the first living cell was studied under the simple microscope by Antony van Leeuwenhoek, right? After this, in 1839, the cell theory was formulated. We are going to discuss in length also, that at length, what the cell theory is and what kind of modifications were done after that. So this cell theory was initially formulated by Matthias Jacob Schleiden and Theodor Schwann in 1839. Okay, after that, this particular cell theory, due to new evidences that had come up, new knowledge about the cell that we now knew, it was modified by Rudolf Virchow in 1855. The major modification that he did was that he stated that all the cells came from pre-existing cells, right? So there was no spontaneous pre-existing cells. Then Walter Fleming described the process of the cell division in 1882. Right. So this is also what we are talking about the 19th century till now. Right. So this was as early as people were interested in cell and understanding the mechanics of how the cell biology works. Right. Now we said that we'll be discussing this cell theory at length, right? So what is cell theory? A cell, the cell theory states that a cell is a unit of structure, physiology, and organization in the living things. The cell can have a distinct identity or be a building block in the construction of multicellular organisms, right? So, for example, if there is a unicellular organism, that one cell is an entire organism, right? So it will have its distinct identity. But in case of us, for example, our cells function as, function in masses or they function as tissues, right? So a single cell in our case will not be that I'm sorry. Yeah, a single cell in our case will not be that relevant or that, they function, similar kinds of cell come together, they form tissues, and that is how they basically drive functions in our body. Then cells form by free cell formation, similar to formation of crystals. That is spontaneous generation. So this is the line that was said when Theodore and over the two scientists when Theodor Schwann and Jacob Schleiden actually proposed cell theory, right? So out of this, we know now that this third statement is definitely incorrect. There is no spontaneous generation of cells, right? The cells have to come from pre-existing cells. So the initiation of this, amendment in the cell theory was done by Rudolf Virchow, as we talked about. And the modern cell theory now states that the cell is a fundamental structural and functional unit in the living beings. All organisms are made up of one or more cells, and the cells arise from pre existing cells by cell division. Also, cells carry hereditary information that is passed on to daughter cells during cell division. All cells have similar chemical composition and structural organization, and energy flows within the cell, right? Also, when we talk about cells, there are two major kinds of, cell morphologies kind of that occur, and they differ between these two groups, that is prokaryotes and eukaryotes, right? So in the prokaryotic cell, the cell organelles will have a different kind of arrangement, while in eukaryotes, the cell organelles will have a different kind of arrangement. That is what is meant. So this tree over here basically shows that we all have arisen from a universal common ancestor, right? First, maybe during evolution came the bacteria, which are the part of this class that is prokaryote. So they are unicellular and they do not have a well-defined nucleus, right? So we'll discuss what is different in prokaryotes and eukaryotes later. Also, during evolution, the archaea branched out, which are also prokaryotes, but they are a little different from bacteria and they are closer to the eukaryotes than the bacteria, right? And eventually, the eukarya arose. In eukaryotes as well, we have unicellular organisms such as Monera and multicellular organisms such as our cells, right? When we talk about a prokaryotic and eukaryotic cell, there are some distinctive differences that you will find, right? So prokaryotic cell can range up to zero point 0.1 to 10 micron, while a eukaryotic cell can range from 10 to 100 micron, right? Also, over here, you will see that the prokaryotic cell often have some kind of flagella or maybe some kind of cilia or feet-like structures that is known as podia that are often present in these cells. Why is that so? Because this is a single cellular organism, right? And a single cellular organism needs to survive, and one of the survival mechanism is movement, basically, or locomotion, right? So if there is danger, this particular organism has to move away from it. If there's food, this particular organism has to move towards that, right? So these structures are often present in prokaryotic cells, which are absent in eukaryotic cells, right? Apart from this, you can see that both of them do contain the plasma membrane, the cytoplasm, and the DNA. But in case of eukaryotic cell, you can see that this DNA is inside a defined cell organelle, which is known as a nucleus, right? While in case of prokaryote, there is no defined cell organelle that is present. The DNA is present as such in the cytoplasm, has a mass in the cytoplasm, and this is basically known as nucleoid, right? So this does a prokaryotic cell does not have a well-defined nucleus. That is, it does not have any nuclear envelope, nuclear wall, or any separation of the nucleus from the cytoplasm. Right. Apart from that, we can see that both of them again do have ribosomes, right? And cell organelles, if you see, there are a lot of cell organelles that are present in eukaryotic cell, while in prokaryotic cell, the number of cell organelles are very limited, right? You can see a nucleoid, you can see some ribosomes in the cytoplasm, and that is it, right? And there's no mitochondria that can be seen. There's no endoplasmic reticulum that can be seen. So that is another key difference that is present. When we talk about differences between prokaryotic and eukaryotic cell, we can see that prokaryotic is unicellular, while eukaryotic could either be unicellular or multicellular, like I said earlier. These prokaryotic cells range in size from 10 to 100 micron, while eukaryotic cells range from 0.2 to 2 micron. Now, okay, although this is something that I have picked from ma'am's slide, but the previous one I had also picked from course in NPTEL. So these are two contradicting data. I did not ponder upon that earlier. So we'll just check that, okay? And if I miss that, please go and check what is the exact size range and which one is bigger, right? Then in prokaryotes, there is nucleoid that is present. That is, there is no defined nuclear membrane. While in case of eukaryotes, a nucleus, which is membrane-bound compartment, is present in the cytoplasm, and the DNA is bound within this nucleus. Okay. In prokaryotic cells, the DNA is circular, while in eukaryotic cells, the DNA is linear. Prokaryotes have cytoplasm with ribosomes, while eukaryotes have cytoplasm with ribosomes. Along with this, there are other organelles on which the ribosomes are bound, right? These are membrane-bound ribosomes. They are present on lysosomes, Golgi, ER, peroxisomes, etc. In prokaryotes, again, the mitochondria is absent, so is the centrosomes, while eukaryotes have both the mitochondria and centrosomes present. Examples of the prokaryotic cell include eubacteria and archaebacteria, while eukaryotic cells include animals, plants, yeasts, fungi, protists, etc. This is again a revision of what are the kinds of cell components that are present in case of prokaryotes versus eukaryotes. So prokaryotes have nucleoid, cytoplasm, ribosome, plasmid, uh, plasma membrane, cell wall, and pili or cilia or flagellum that is present, right? While eukaryotes have nucleus, cytoplasm, ribosomes, Golgi complex, endoplasmic reticulum, mitochondria, lysosome, plasma membrane, cell wall, and plastids, right? Now over here, this is one thing that we did not discuss earlier, that prokaryotes, apart from their major or genomic DNA, they also have plasmid, right? This is also a kind of DNA that is extra genomic DNA that is present within these organelles, within these organisms, right? Also, now if we come to prokaryotes, we largely distinguish these bacteria that are present within this class of prokaryotes as either gram positive or gram negative, right? So now what is the difference between these two and how can we actually distinguish them, right? So in case of gram positive bacteria, what happens when we do gram staining? They show a purple pinkish bright color, while gram negative bacteria do not stain like that, right? Now, what is the difference between the two structures that actually leads to this difference? In case of the gram positive bacteria, as you can see, it has an inner cell membrane, right? Outside of that is the periplasmic space in which certain lipoproteins are present, and outside of that is a thick peptidoglycan layer, right? And this peptidoglycan layer is what is stained by this gram stain, right? So when this gram stain is given, since the peptidoglycan layer is exposed and it is pretty thick, it gives a dark pinkish purplish color, right? And these are the gram positive bacteria. While in case of the gram negative bacteria, there is only a difference in the outer coat, right? So it also has an inner cell membrane. Outside of that is the periplasmic membrane, and it also has peptidoglycan. But as you can see, the peptidoglycan layer is very, very thin, right? It is not as thick as that present in gram positive bacteria. Along with that, outside of this is another periplasmic space and then an outer membrane. So gram positive bacteria completely lack this outer membrane, right? While gram negative bacteria consist of this additional outer membrane and a very thin peptidoglycan, due to which actually the gram stain cannot actually penetrate and stain these particular kind of bacteria. That is why the distinguishing this feature arose that the ones that have exposed peptidoglycan thick layers are gram positive bacteria, the others are the gram negative bacteria, right? And also due to the presence or absence of this outer membrane, they also differ in their virulent properties, right? So there are some bacteria that can infect us or other higher eukaryotes, right? So their virulence is also different based on whether or not they have an exposed peptidoglycan or an outer membrane that is present or not. Okay. Also, we have talked about the cell components both in eukaryotes and prokaryotes. Now, there is an additional cell component that is present, right? So, this cell component is majorly found in the eukaryotes, right? It is known as cytoskeleton. So cytoskeleton is composed of multiple other substructures, which are microtubules, intermediate filaments, actin filaments, and centrosomes, right? And this cytoskeleton is very, very important because it helps the cell maintain its structure, right? Along with it helping the acting as a skeleton and helping the cell maintain its structure, it is also very key in processes such as DNA replication and cell cycle division, because all these constituents of cytoskeleton, that is actin and intermediate filaments, microtubules, centrosomes, all of them have some role to play in cell cycle divisions as well, right? Then now that we have also talked about prokaryotes and eukaryotes, we can talk about the two or highest order eukaryotes that are present. One is the plant cell and the other is the animal cell, right? So the animal cell has one kind of general basic structure, and the cell has a different kind of general basic structure. It does not mean that all plant cell definitely look like this or all animal cells definitely look like this, right? There could be some differences between tissue types, but generally speaking, most of the animal cells have this kind of a structure and most of the plant cells have this kind of a structure, right? So as you can see, these cells look very, very distinct, right? Although they have a lot of commonalities that are present between them. For example, ribosomes are present in both the cases. ribosomes, lysosomes, nucleolus, mitochondrion, rough endoplasmic reticulum, smooth endoplasmic reticulum are all common factors, right? Now, apart from this, there is peroxisome that is present in both, Golgi apparatus that is present in both, and cell membrane that is present in both, and cytoplasm, right? Apart from this, you can see that the vacuole that is present in case of the animal cell is much smaller, or sometimes it is not even there. But that is present in case of the plant cell is very huge, right? Or sometimes what happens is that it covers uh, almost the entire cytoplasm, right? So this is still not as huge as it is normally found in case of plant cells. So that is one key difference. Now other differences are that this kind of cytoskeleton elements such as centrioles, microtubules are only present in the animal cell and not in the plant cell, right? Whereas there are some structures such as cell wall that are present in the plant cell only, right? Cell membrane is present in both, but plant cell outside of the cell membrane also has an additional membrane which is known as cell wall, right? And it is a rigid structure. Uh, Apart from that, there are some plastids such as chloroplast, amyloplast, and other kind of plastids that are present in plant cell but not in animal cells, right? Why? Because these plastids in plant cell are basically responsible for either aiding the plant cell to manufacture its own food or to store its food. That is done by different kinds of cells in animal cells, and in animals the food is not prepared, right? Prepared in the sense that the food is not auto-synthesized, right? We utilize some energy from the food that we intake, right? And that is in the form of various kinds of molecules, NAD plus, ATP, etc., right? Okay. Now, having talked about the prokaryotes and eukaryotes, there is a hypothesis of how basically the various cell organelles and eukaryotes would have arisen, right? And this hypothesis is known as endosymbiosis hypothesis, right? What does this hypothesis say? That initially there used to be an archaea, right? And this archaea would have derived anaerobic ancestral line with primitive nucleus, right? So after that, it could have happened that there was a different bacteria or bacterial ancestor of the mitochondria, which is aerobic eubacterium. Now, since this one is an anaerobic ancestor, it might have, formed a symbiotic relationship with this aerobic bacteria, and that is how basically the mitochondria in was kind of evolved, right? And initially, it might have looked more like a bacteria, but now in plant and animal cell, it looks more like this with cristae and inner and outer membrane. Now, why it is also believed that this might have happened is one, because mitochondria has its separate set of DNA, right? So there is DNA in the nucleus as well, right? Apart from that, there is separate DNA that is present in mitochondria that is basically represented over here also, right? Apart from this, you can see that like the cell that has a double membrane structure, mitochondria is also one of the organelles that have a double membrane structure. It has an inner membrane and an outer membrane. Sorry. Yeah. So after this, this is how the animal cell would have arose. And in case of the plant cell, there would have been an additional bacterial ancestor, that is a cyanobacterium that might have, again come into a symbiotic relationship with this existing nucleus and mitochondria containing ancestor, and that is how plastids or chloroplast would have been formed, right? So that is how a plant cell might have been formed. So this is the endosymbiosis hypothesis, right? And it is based on some key features, like I told you, that these are organelles that have double membrane structure. Mitochondria has this thing, DNA of its own, etc. Right? Then we come to model organisms. Sorry, just give me a moment. I'll just have. Yeah. So when we talk about model organisms, there are various kinds of model organisms that are present. They could either be plants or they could even be bacteria, yeast, etc. Apart from that, there are certain invertebrates that are, used as model organisms. They are C. elegans, Drosophila, and certain vertebrates are also used as model organisms, which are zebrafish, xenopus, chicken, mouse, and human. Right. So, what are model organisms? For the study of any disease or pathology or even natural processes that occur within us, we need, you know, some organism to study it on, right? It is not like I will look at you and I will suddenly think of, mitosis happening or mitosis occurring in you, right? So that for that we need simpler organisms on in which we can actually observe that, okay, this thing is occurring, right? So for that purpose itself, there are model organisms. Why are we listing humans also as model organisms? Because there are some, for example, brain related studies in which there are tests that are conducted, questionnaires that are floated, then games that people or participants are asked to play, and then they have to answer another set of questions. Then they are, their physical, the medical history is taken, and based on that, some observations are made for an entire population, right? So even if there is not, physical experimentation that is going on with you, there could be other kinds of or other forms of experimentation that could happen, right? For example, even in mice, it is not necessary that the mouse will always be sacrificed and then its tissues will be harvested, then it will be looked at, and then only we will see what is going on, right? Sometimes what we do is just to assess a condition, if there is a genetic model of a mouse or a a model of a mouse, we will just assay its behavior to see that, okay, I think that this might be the problem with it. Only if you have to go into molecular levels will you actually have to, physically work on this particular model, right? So there are multiple kinds of model organisms that are present, and you can always read more about it. Apart from this, these are a few points that were discussed in ma'am's slides. So since we were talking about cells and how cells are made, there are some essential molecules that are present in virtually all cells. But if we talk about elements in the human body, there are some common elements such as carbon, hydrogen, nitrogen, and oxygen. These atoms are linked by covalent bonds to form molecules. 99% of the human body has these elements or is made up of these elements only. There are other elements which are the macronutrients, as we call it. They are the sodium, potassium, magnesium, calcium, phosphorus, sulfur, and chlorine, right? And again, yet again, there are trace elements which constitute only 0.1% of the human body, which are the micronutrients. And despite that, they are very, very important, right, for function, because they could be the, cofactor of some particular enzyme, or I'm sorry, I just had a very long day. Uh, these micronutrients are, minerals such as, or nutrients such as fluorine, iodine, okay. these are the micronutrients that are actually present, right? So there is a list of them. You can go through them. Then when we talk about the cell composition, about 70% of the cell composition is also water, right? Like our body is 70% water. So that is a very, very good correlation, right? So it is just, extrapolating what is there in the cell to the entire body. And these water molecules have a very, very characteristic feature, which is known as hydrogen bonds, right? So they have the electropositive H ion, which is shared by two electronegative atoms. And this is a characteristic feature of water, right? Then there are some organic molecules, which are small molecules such as sugars, fatty acids, amino acids, and nucleotides, which are basically the building blocks of these macromolecules or large molecules that are large polysaccharides, lipids, membranes, proteins, and nucleic acids, right? So these small molecules are essentially the building blocks, and these are the final products. There are other ions such as or inorganic ions, and the common ones being sodium, potassium, magnesium, calcium, and chlorine. Then the molecules bind to each other by non-covalent bonds. These non-covalent bonds could be electrostatic or ionic bonds, hydrogen bonds, van der Waals attraction, hydrophobic force of water, which pushes non-polar surfaces out of the H-bonded water network, right? Then, even at this cellular level, there is some integrity of atoms, molecules, and bonds that is maintained, right? Speaking chemically, is the smallest particle that retains the properties of an element, right? There are various examples of these atoms that are given. When we talk about reactivity of these atoms, the electrons in these atoms will determine the chemical behavior of that is present in them, right? Electrons can exist only in certain discrete states, which are known as orbitals. So these are the orbitals, these rings that are present. Each can accommodate only a certain number of electrons, right? So, for example, we know that the innermost orbit can only hold two. The outer one can hold eight. After that, again, eight, then 18, 18, right? So, we have studied this in chemistry very early on in science, right? Then an unfilled electron shell is less stable than a filled one. So we know that if there is one electron in this particular inner shell, it is unstable. If there are two that is present, so it is full. So it is very stable, right? So for example, if eight are present in the outer shell, it is again very stable. But if seven are present or five are present, it is not that stable, right? Thus, the noble gases are very stable because they have eight or 18 out on the outer or valence shell, right? Then atoms with an incomplete outermost shell have a tendency to interact with other atoms to complete the shell. This can occur by transfer or sharing of electrons. This number of electron is known as valency, right? So these are some of how methane looks. It is a symmetry of how basically these atoms are shared between these hydrogen and the carbon that is present, right? It has this tetrahedral symmetry. We are not going into details of this. Also, when atoms form covalent bonds with other atoms, the bonds have definite orientations in space relative to one another. The orientations of bonds reflect the orientation of orbits of the shared electrons. Hence, the covalent bonds between multiple atoms are characterized by specific bond angles. There are also special bond lengths and bond energies, right? So a lot of class 11th chemistry, I think you guys might be recalling. So when we talk about bonds, there are some major kinds of bonds that are present. When we talk about covalent bond, it is one of the strongest bonds that is there. It could be a single covalent, double covalent, triple covalent, or polar covalent bond. So in single covalent, there's a sharing of two electrons, right? In double, there's a sharing of two pairs of electrons. Triple, there is sharing of three pairs of electrons. While in polar covalent, when the atoms of two different elements form a covalent bond, the two atoms usually attract the shared electrons to different extents, right? So there will be the sharing is not equal in this case, right? There is more utilization of those electrons by one atom and less utilization by the other atom. The pair of electrons are shared unequally with a partial transfer between the atoms, resulting in a polar covalent bond, right? Then non-covalent bonds important for intermolecular interactions in the cell. The non-covalent bonds include hydrogen bond, ionic bond, van der Waals attractions, and hydrophobic force. So hydrogen bond again is a very, very key kind of bonding that is present in nature as well as in living beings. So hydrogen bond is an interaction between the electropositive hydrogen atom is partially shared by two electronegative atoms. Directional strongest when all three atoms are in the straight line. Water weakens these bonds by forming competing hydrogen bonds. Okay. And then there is ionic bond when electrons are transferred from one atom to the other. Now, this was another very basic kind of bonding that we had studied very early on, right? And then van der Waals attractions. Now, these are weak bonds that are present. So, attraction between two neutral atoms at close range arises from formation of induced dipoles. Fluctuation in the electron cloud produces a transient dipole that induces a oppositely polarized flickering dipole in a nearby atom. Very weak interaction, but not weakened by water. So it is very weak by itself. Okay. Also, when we talk about the hydrophobic force, it's caused by pushing away of nonpolar groups out of the hydrogen bonded water network, non-specific, and it is important for the protein folding, right? So, yeah, all of these are actually very, very key in protein folding. Just let me view the lobby. Somebody's trying to join in. Yeah. Okay. Yeah, so we had discussed this. Then we talk about water. So we have talked about the kind of bonds that are present, right? In case of water, we know that it has polar covalent bonds. That is, it forms a dipole. So we have already studied what is a polar covalent bond. That there is unequal sharing between atoms, right? Some atom is going to take more portion of a certain thing, and another one is going to take less of it. For example, if in a very, very ideal situation, if you and your sibling are present, you will want to, if there's a pizza with 4 slices, you will want to have two and you will give that your sibling two, right? But sometimes it might happen that maybe your sibling is very hungry, you just ate, or maybe you are very, very generous and you want your sibling to have the entire pizza, or you want them to have 3 pieces, you will only take one. So this will be a scenario of polar covalent bond, right, where we are sharing, but it is unequal sharing. Then, in case of water, it has a net neutral charge, but electrons are asymmetrically distributed. The oxygen nucleus draws electrons away from the hydrogen nucleus, which impacts positive charge to hydrogen. Also, excess electron density to oxygen atom creates weak negative regions with tetrahedral symmetry. Then hydrogen bond is basically an interaction between hydrogen with a partial positive charge that shares a polarized covalent bond with a more electronegative atom. So this one will be the acceptor with a atom with a partial negative charge, which is going to be the donor. That is part of another dipole. Okay. So due to polarized nature, water molecules can form hydrogen bonds. Water molecules can form a hydrogen bonded lattice. They are weaker than covalent bonds, and the strongest hydrogen bonds are strongest when 3 atoms are in a straight line, right? Water molecule can bond with 4 others to form a transient flickering cluster. So again, I actually did not want to discuss this bonding part in detail, but okay, we'll read this also. So there are some small organic molecules like we just talked about. They are molecules such as sugars, fatty acids, nucleotides, amino acids, and other building blocks of macromolecules, right? So other than water, most molecules in cell are based on carbon. Carbon can form large molecules. It is small, has 4 electrons and 4 valencies in its outermost shell. Hence, it can form 4 covalent bonds with other atoms. A carbon atom can also join to other carbon atoms via highly stable covalent carbon-carbon bonds, and it can form rings and chains and can form large molecules with no obvious upper limit to their size. These carbon-based compounds in cells are referred to as organic molecules, and various chemical groups that are reactive are also found in biological molecules. Example are hydroxyl, carboxyl, amine, and phosphate. Right? There are, this is just a brief representation of the 20 amino acids that are present or that actually form the proteins, right? And the side chains of these amino acids vary, and based on their side chains, they are either non-polar or polar uncharged or acidic or basic, right? So these are the kinds of this is a single a letter representation of these amino acids, right? And based on that, you can remember like which ones are non-polar, which ones are polar uncharged, which ones are acidic, which ones are basic. Then coming to macromolecules, most of these are polymers, right? They are very abundant, and the information regulatory and as well as structural roles are played by these particular macromolecules. Um, yeah. The examples of such macromolecules include polysaccharides, nucleic acids, and proteins. Proteins are polymers of amino acids. They are formed by a condensation reaction and by loss of water molecule and linkage via a peptide bond. Right? So this could be a primary structure of a protein wherein there are multiple amino acids that are joined together. Then in a secondary structure, you will see that there will be further coiling of this, or there will be some kind of sheets that will be formed so that this very long polypeptide structure that is forming will get compacted. Then will come the tertiary structure. Now, in this tertiary structure, there will be further kind of coiling or some further kind of, sheet formation that will occur. And what will this coiling do? That it will give a structure to this protein, right? Now, if it is a structured protein, it might be relevant for that regard. If it is a receptor protein, it will have to have a structure such that it can recognize the ligand that it is determined to recognize. If it is, for example, enzyme, then it should have some site which can recognize the cue that okay, now this enzyme has to work, right? So that is what will be done in this tertiary structure. Most proteins have until this tertiary structures, but sometimes especially in case of enzymes, there are multiple subunits that are present. So these subunits, when they come together, so when different tertiary structures come together and they form a subunit, it is basically known as a quaternary structure. Okay. So the proteins have remarkable properties and functions. They fold via non-covalent interactions into precise stable shapes, crucial for their functions. They can also associate with the other macromolecules via non-covalent interactions to form molecular machines performing complex tasks. After this lecture came the talk about metabolism, right? So what is metabolism? It is basically the chemical reactions in the body cells that change the food into energy. So whatever macromolecules or whatever small molecules that we are actually getting in our body, it is coming from somewhere, right? We are not synthesizing them on our own, right? Macromolecules we are synthesizing from small molecules, right? But where are the small molecules coming from? They are coming from the food, right? So this change of energy from this extraction of energy from food is known as metabolism, right? In a cell, the chemical reactions occur in aqueous environment in a dynamic network of water molecules bound via hydrogen molecules. Chemical reactions within the cell generate and also release heat. This synthesis requires energy that comes from oxidation of food and organic molecules. When we talk about metabolism, there are many different reactions that are constantly being performed in a living cell. They could majorly be categorized into under two categories. One is the anabolic or the biosynthetic reactions, and the other is the catabolic or degradation related reactions, right? So, anabolic reactions use small molecules and energy to synthesize other molecules, while catabolic reactions break down food into smaller molecules and generate energy. These can be utilized for biosynthesis, right? So, synthesis of other things can be done by breaking down some things and utilizing that energy, right? Then we talk about activated carrier molecules. So, what are these activated carrier molecules? They are produced by oxidation of food molecules and they store energy as chemical bond energy in the form of energy rich covalent bonds. This can be used for energy requiring reactions or biosynthesis. Example ATP, NADH, NADPH, FADH2, acetyl coenzyme A, carboxylated biotin, S-adenosyl methionine, uridine diphosphatase glucose, etc. Right? Then ATP out of this is the most common energy currency and it is kind of preserved across species also. That ATP is generally the energy currency in most of the organisms, right? ATP hydrolysis is coupled to synthesis of biological polymers. ATP is also a building block of nucleic acids, right? So when we talk about ATP, it is the energy currency or fuel of the cell. It is formed via reactions driven by energy release during oxidative breakdowns of food. Its three phosphates are linked in series by two phosphoanhydride bonds. Breaking these bonds release large amount of useful energy, right? It is ATP, triphosphate. If by breaking each of these phosphate bonds, there is a large amount of energy that is released, right? Which is why it is known as the energy currency because energy stored in this. The terminal phosphate group is often split off by hydrolysis, sometimes transferring a phosphate to the other molecule and releasing useful energy that drives energy requiring biosynthetic reactions. There are some chemical bases for these large negative standard free energy of hydrolysis of ATP, and this I have discussed at length in the practice sessions, and so has ma'am, right? So I am not going to repeat that right now because I don't think we'll be able to get done with this revision of half the course also then. Then we come to enzymes, right? So what are enzymes? They are biological catalysts and they accelerate particular reactions within the cell. So there are some things within the cell that will naturally occur, but they will naturally occur at a very, very slow pace, right? So enzymes basically act as catalyst or they increase the efficiency of this process. They are commonly proteins, although there are some RNA catalysts which are also termed as ribozymes, which also exist. Enzymes bind specific substrates in a way that lowers the activation energy of their needs to occur. Enzymes have much higher affinity for unable transition state than for the stable form of substrate, and it binds lower its binding lowers the energy of transition state. Okay, so the same thing that anyway this transition was going to occur, but the threshold for transition is lowered when the enzyme is in action. Okay, enzymes are highly specific and they catalyze only a specific type of reaction. Then we talk about coenzymes. So coenzymes are basically small organic molecules that work with enzymes to enhance reactions rates. Okay, so enzyme is also acting as a catalyst. This is further enhancing reaction rates. It is recycled to participate in multiple enzymatic reactions. Examples are NAD positive, then hydrogen, etc. Okay. Then, if we talk about a living cell, it is a complex system that is bounded by membrane that performs complex processes that are purposeful and to maintain itself, right? Also, it will have minimal self-replicating. It is a minimal self-replicating unit of living matter. But the question that arises is is a cell just a bag of chemicals that react? So no, this is not true, right? The reactions that go on are ordered and they are highly organized. The processes follow established chemical and physical principles. Then the processes are interconnected and purposeful, so they are always solving a purpose, right? It is not that okay, this is not required, but I will keep on making this particular substrate. So that is not how it works. And the cell can grow and replicate itself, right? One of the other integral components of the cell was the plasma membrane, right? So the plasma membrane, which is also known as a cell membrane, is a membrane that is found in the cells that separates the interior of the cell from the outside environment. There are certain components that are present in the plasma membrane. The major components include phospholipids, glycolipids, and cholesterol. The phospholipid consists of two hydrophobic fatty acid chains, which are linked to two carbon atoms in glycerol. Third carbon atom of glycerol is bound to phosphate group, that is in turn attached to small polar molecule, example choline, serine, ethanolamine, or inositol. There are certain examples of these phospholipids, right? Then glycolipids have two hydrocarbon chains linked to a polar group that contains carbohydrates. Then cholesterol has four hydrocarbon rings that are strongly hydrophobic. One end has attached hydroxyl group that is weakly hydrophilic. They are also amphipathic molecules, so they are kind of in between hydrophobic and hydrophilic. They form self-sealing lipid bilayers, and the polar head groups are exposed to water, and hydrophobic tail face each other in the interior of the bilayer. So this is also a key feature that we will study in the fluid mosaic model later, right? So this is what the plasma membrane looks like, right? There is asymmetrical distribution of phospholipids. So outside on the outer the cytosolic side of the plasma membrane, there is sorry, cytosolic is this side. On the outer side, yeah. So this is the cytosolic side and this is the outer side, right? So on the outer side of the plasma membrane, there is an abundance of glycolipid, phosphatidylcholine, sphingomyelin, and cholesterol. While on the inner side, phosphatidylserine and ethanolamine are the major constituents of this particular inner layer of inner side of the plasma membrane. There are also some phosphatidyl inositol, some cholesterol, and some minute levels of sphingomyelin and phosphatidylcholine that are also present. And this asymmetry is actually functionally important. So we will study this much later when we will understand that this is how, our cell is also recognized. So during autophagy, for example, what happens is that, or during apoptosis, what happens is that there is an eat me signal that is find me and eat me signal that is present, in which this phosphatidylserine and ethanolamine, which are supposed to remain inside, basically protrude out. That is how the surrounding macrophages know that, okay, this cell has something wrong with it. So we need to engulf it, right? So this asymmetry is definitely functionally important. Also, there is a mobility of phospholipid molecules in the bilayer, right? So they can move within the bilayer. It is not a fixed structure. Now, this movement is usually lateral. So they can move like this, side by side. It usually, and these are the two layers. So they can move like this. But more often than not, it does not happen that they will flip, right? If the flipping is occurring, it is very, very rare. And usually it is not the case, right? Then when we talk about membrane proteins, right? So this was the plasma membrane. There are some proteins that are associated with them. They could either be integral proteins or peripheral proteins. In case of integral proteins, they are embedded within the membrane, right? In case of peripheral proteins, you can see they are on the top of the membrane that are present, right? So integral proteins are transmembrane proteins. They have one or more membranes spanning alpha helices with 20 to 25 non-polar amino acids or a beta barrel. They have exposed hydrophilic portions on either side of the membrane, right? While peripheral are not inserted into membrane but associated via interactions with integral membrane proteins or with phospholipids. Then we talk about this very, very key or very, very important model, that was the fluid mosaic model that was given by Singer and Nicholson. The fluid mosaic model is the most widely accepted model of how a plasma membrane looks like and how it functions. So it says that the overall membrane structure is determined by combination of hydrophobic and hydrophilic interactions. The globular integral membrane proteins are inserted into phospholipid bilayers. These membrane spanning proteins are amphipathic and with non-polar regions inserted in the bilayer and polar regions exposed to aqueous environment on either side. The membrane proteins can move laterally through the lipid. So this is all that we have discussed earlier also. After that, there were some updates that were done or some modifications that were brought about to this original Singer and Nicholson version of a fluid mosaic model. And the updated version says that mobility of many membrane proteins is restricted in contrast with free movement in bilayer as it was proposed earlier. The membranes are composed of distinct domains that have important structural and functional roles. Some plasma membrane proteins are associated with underlying cortical cytoskeleton like spectrin and actin that restricts their mobility. They can also act as barriers to restrict the mobility of other proteins. Also, cholesterol, sphingomyelin, and glycolipids cluster to form semi-solid lipid rafts that compartmentalize the plasma membrane into distinct domains, right? They are enriched in GPI anchored proteins and certain transmembrane proteins involved in specific functions, movement, signaling, endocytosis, etc. The caveoli are a subset of the caveoli are a subset of lipid rafts. As a membrane protein, caveolin interacts with the cytoplasmic protein cavin and the cholesterol invaginations, endocytosis signaling or lipid transport, right? So then we talk about transport across the cell membrane. There are multiple ways of transport. Let us just skip this slide. I will come to this slide, which talks about various kinds of transporters. So there could be channels that are present. There could be ion transporters. There could be other kinds of ligand gated there are multiple ways that these channels function, and there could just be pores also that are present, which will help in movement of some molecules, right? So ion transporters actively move ions against concentration gradient. So it means ATP is being used for this transport to occur, and it means that even if this dark blue is more concentrated outside, still this transporter will throw it out, right? While in case of, and it basically create ion concentration gradient. So it is very important for signal transduction actually in cells. So signal transduction is not something that we have dealt with in this course, but it is very, very important, right? For signaling to occur. Then in ion channels, they allowed uh, ions to diffuse down concentration gradient, means it is a passive channel. It means that this dark blue color, ions, if they are more abundant outside, it will allow it to come inside, right? They cause selective permeability to certain ions, right? So if it is a sodium ion channel, it will only allow sodium to go. If it is a potassium ion channel, it will only allow potassium to pass. So there are different mechanisms by which other molecules are inhibited from passing through these channels, right? And it is very interesting to actually look at those mechanisms. But for now, we are moving on to bioenergetics and metabolism. Now, please give me a moment. I'll just put my laptop on charge. So, bioenergetic and metabolism is again how basically various, substrates are being utilized and how it is basically converting into energy within our body. So, there is enzymatic breakdown of food molecules which occurs outside the cells or in the lysosomes. There are digestive enzymes that are present in the lysosomes which break down these proteins into amino acids, polysaccharides, and sugar. Uh, uh, the polysaccharides into sugars and fats into fatty acid and glycerol. So all its components it is breaking down into. So this is what is catabolism. The small organic molecules produced after digestion enter the cytosol of the cell where their oxidation starts. Glucose is converted into pyruvate, which is two molecules of pyruvate via glycolysis. So glycolysis, another very key cycle. Then ATP and NADH are also produced. Pyruvate then passes into cytosol, from cytosol into mitochondria. In mitochondria, each pyruvate molecule is converted into carbon dioxide plus 2 carbon acetyl group, which becomes attached to coenzyme A, forming acetyl coenzyme A by oxidative decarboxylation. The carbon dioxide and NADH are also produced. Um, and the acetyl group is linked to coenzyme A via high energy linkage and can easily be transferred to other molecules. This acetyl group of acetyl coenzyme A is transferred to the 4 carbon molecule oxaloacetate to form citrate and then enters series of reactions called the citric acid cycle or the TCA, right? The two carbon of citrate are oxidized to carbon dioxide and oxaloacetate is regenerated. One molecule of ATP, one of FADH2 and three of NADH are formed from each turn of the cycle. The electrons from NADH and FADH2 are transferred via ETC in the inner membrane of molecular oxygen in the inner membrane in the mitochondrial membrane to molecular oxygen, and it is coupled to formation of ATP molecules by oxidative phosphorylation. Right? This is what happens. First, there is glycolysis that is occurring, which is breaking down glucose into two pyruvate molecules. These two pyruvate molecules are basically forming acetyl coenzyme A and NADH and carbon dioxide. NADH can directly form ATP and through oxidative phosphorylation. In the ETC, this acetyl coenzyme A gets into this Krebs cycle or TCA, and it basically forms more 2 ATP over here itself, then NADH, FADH2, and carbon dioxide, and these will go to the mitochondria. Okay, we will see that later. And they will basically form energy, right? Then we come to the chemiosmotic theory given by Mitchell in 1961. It basically says that there's coupling of phosphorylation to electron and hydrogen transfer by chemiosmotic type of mechanism. So this is a very huge theory. We have discussed that in one line over here. Uh, uh, so this basically leads to synthesis of ATP, which is driven by electrochemical gradient across a membrane rather than a high energy intermediate, right? The intermediate that couples electron transport to ATP synthesis is a proton electrochemical gradient across a membrane. The proton gradient is produced by electron transport, and the flow of protons back to the membrane is in the energetically favored direction is coupled to ATP synthesis. So this is what it looks inside the mitochondria during the electron transport chain. So this is the inner mitochondrial membrane, right? And you can see that within the inner mitochondrial membrane is actually where this ETC or electron transport chain is working. It will either work in 1, 3, and 4 or 2, 3, and 4 disorder, right? And it will throw out protons into the intermembrane space, right? Now, these protons will then enter via this ATP synthase machinery, and when they are entering back, they will convert ADP to ATP, which is the energy containing molecule, right? So, the electron transport chain, the energy derived from breakdown of carbohydrates, fatty acids is converted to ATP by oxidative phosphorylation in the mitochondria. As we have already seen that there is this inner mitochondrial membrane. From within the inner mitochondrial membrane, wherein this TCA cycle has also occurred and FADH, NADH, etc. are already present. They will go, if it is FADH, it will go through these coenzymes 2, 3, and 4. If it is NADH, it will go through 1, 3, and 4. And there will be export of protons into the inner mitochondrial intermembrane space of mitochondria, right? And in this intermembrane space, these protons will be accumulated, and through the ATP synthase pump, they will be thrown back into the inner mitochondrial membrane, into the mitochondria through this membrane, and this will drive the conversion of ATP to ADP to ATP. Yeah, Deepti, did you want to say something? I missed that.
Yes, ma'am. Ma'am, I think PPT is not visible. The slides are not visible to us. Is it so for me or everyone? I don't know, but I cannot
I,
see the presentation.
I did not know that. I'm so sorry. Um, I'll just share it again. Okay, I
Yeah,
can
it's
see the
visible
pop-up
now.
now. I hope it's visible.
Thank you. Yes, ma'am, it
Okay.
is visible. Thank you.
Okay. Then we come to the discussion of genetic material, right? After we have discussed how energy is, how metabolism is occurring, how energy is being utilized. After that, we also need to know that because one of the major cellular components is the nucleus, which is containing the genetic material, we need to know a little bit more about the genetic material and what it is, right? So ideally, what should be a genetic material, right? So but when a rationale was proposed for it very long ago, then it was deemed that it should be able to provide a rationale for faithful inheritance of traits, for evolution, that how do selectable variations arise, and there should be a possible mechanism for this to occur, right? So if we at that time it was deemed that if they found a genetic material, it should be able to answer these questions, right? So, by light microscopy, chromosomes were observed to appear in dividing cells, and be segregated equally during cell division. Right. So it was assumed that these chromosomes have the genetic material. But now the query was that the chromosomes had both DNA and the proteins. Right. So we did not know which one is the genetic material. So, when this quest was on, the contenders for the genetic material were proteins and DNA. And since there is also RNA that is present as in many of these, organisms, RNA was also another contender, right? And it was seen that if there is this transfer of genetic material between species, even the non-virulent strains of this Streptococcus pneumonia, the smooth strain was virulent. That is, it would kill the mice. It was infect and kill the mouse that it was infected by this particular strain. And the R strain or the rough strain of the Streptococcus pneumonia was not virulent, right? So the mouse would survive. And it was seen that sometimes what happened is that even if this R strain was present and there was heat killed S strain that was given, it means that the strain will definitely have died. But if it is heat killed and it is given to R strain, somehow it transformed the R strain into a virulent kind of strain, right? And when this mouse was studied, it was and the bacteria was isolated, it was seen that S type bacteria was present. It means that there was something within this that it transferred to our strain, right? That could have been the genetic material or the transforming principle as it was then called, right? Now, to understand this, there was another set of experiments that were done by MacLeod, Avery, and McCarthy, in which they have basically, taken various subunits of these materials. Either they have only taken RNA or only protein and only DNA or lipid or carbohydrate, and they have seen that if only this part is segregated out of this S strain and it is given to the R strain, can it transform the cells into S strain? And it was seen only in case of DNA it could do that. That is how we came to know that DNA is indeed the genetic material, right? When we talk about DNA now, which is our genetic material, it is a double helix structure with sugar phosphate backbone on outer side, on the outside, and bases on the inside. There are two strands that are antiparallel and complementary with specific base pairing by hydrogen bonds that is present. Right? We can also see that it has this phosphate group that attaches on this particular hydroxyl group base that attaches on this particular hydroxyl group, and it is 2 deoxyribose, right? Why? Because at this particular location at C2 position, it is just a hydrogen that is present and not a hydroxyl group. So it is a deoxyribose. There is another, this is one of the key differences between DNA and RNA, because RNA over here also has a hydroxyl group and it is a 2 oxyribose sugar or ribose sugar only, right? It is not 2-deoxyribose. So this phosphate group is what will go and attach to this end, that is 5 dash end, and on the 3 dash end, these bases will go and attach. Right? There are different kinds of bases that are present. They are cytosine, guanine, thymine, and adenine. Right? Adenine and guanine have a 6+5 ring structure, while pyrimidine bases such as cytosine and thymine have a six-membered ring structure, right? So guanine and adenine are known as purines, while cytosine and thymine are known as pyrimidines. Also, as you will see later, okay, I'll show that in the next slide maybe. So when we talk about DNA, we talk about a backbone of DNA. Now, how is this backbone formed? It has various kinds of bonding that goes on, right? So at this C1 position, where we said the base is going to attach, there is N-glycosidic linkage between the base and the deoxyribose sugar, right? And there is a O glycosidic bond that is present between the phosphate and the deoxyribose sugar. Two between the two this thing deoxyribose is present, only this phosphate group is present, and between them is the phosphodiester linkage, right? Apart from that, there is a pyrophosphate that is present, which basically gets detached. Two phosphates out of get detached whenever a trinucleotide is being added. Two phosphates get detached and only one phosphate is forming the ring, right? And the deduction or detachment of this pyrophosphate will also actually produce energy, right? So when we talk about, this is what I wanted to talk about in the previous slide, that is base pairing. So there is some kind of unique, pairing in DNA bases that occur. So cytosine will always go and pair with guanine, thymine will always go and pair with adenine, and between cytosine and guanine there will be this triple bond formation, while between thymine and adenine there will be a double bond formation, right? So each base has unique location of hydrogen bond acceptors and donors based on the groups that are attached. Base pairing is specific. That is, one purine will attach with one pyrimidine, and over here G attaches with C and A attaches with T, right? Also, there is a rule that is known as Chargaff's rule, which says that the number of cytosine and guanine are going to be same in DNA, and the number of thymine and adenine are going to be same. Why? Because um, they are going to attach with one another, right? So that is why their number is going to remain constant. This is what the molecular structure of DNA looks like. So there are two sugar phosphate backbones that are running anti-parallel to one another, right? Then DNA is a polymer of four nucleotides, that is bases that are linked to phosphorylated sugars. The nitrogenous bases are purines and pyrimidines, like we have discussed there, and there is phosphate group that is attached. DNA is a double helix, and there are two anti-parallel strands that are present. The purines pair with pyrimidines of other strand via hydrogen bonds. There are two hydrogen bonds between E and T, three between G and C. And if we talk about the B form of DNA, then these are the key features for this particular form. And we have talked about other forms of DNA also that are present in the extra sessions or the practice sessions, right? So you can go and refer that. Then there are definitely DNA needs to be replicated, right? So there would be a mode of replication of DNA. When this mode of replication was not known, there were three speculations that were there, right? One speculation was a semi-conservative model, which said that there would be a parental strand and each parental strand will split, and after splitting it will synthesize one daughter strand on it, right? And that is how replication will keep occurring. Then there was this conservative model, which basically said that the two parental strands will be together. Even if they split, they will form the daughter, each form daughter strands or new strand, and then these daughter strands will go together and the parent strands will come back. And that is how the replication will occur. And then there was a third model, which is known as a dispersive model. What did this model say? That at different parts in a strand, DNA will randomly disappear, right? And then the disappear or DNA will randomly break off, right? This broken off part will synthesize the part that is not there, and the other part will also synthesize the part that is not there, and that is how a replication in DNA will occur. So out of this, now we know that the semi-conservative model is actually the correct model. We will look at that experiment also in a while. Before that, let us talk about how genetic information is coded and decoded, right? So DNA stores genetic information within the sequence of nucleotide building blocks. A gene is a basic unit of heredity, and a sequence of nucleotides that encode synthesis of a gene product is either RNA or protein. Then there is evidence for information flow from genes to amino acid sequences of proteins, right? One of the example is inherited disorder, that is sickle cell anemia, which is linked to S allele of the beta globin gene. Right. So over here we know that the SS individuals that have two of these S alleles will have severe anemia and sickle-shaped RBCs. Sickle hemoglobin differs from normal hemoglobin by a glutamic acid to valine substitution at position 6, right? This basically implies that S allele of gene codes for this change in beta globin protein sequence. Other types of anemia were also associated with single amino acid substitutions in the specific amino acids in or I think there's something wrong in this particular word. In the beta globin chains of hemoglobin. These findings basically indicated that genes or DNA could somehow determine the sequence of amino acids in a polypeptide chain, right? So there is definitely information flow from DNA to the protein. Now the question that arose was how this information flow occurs. Now, why did this question arise? Because DNA cannot be a template for directly ordering the amino acids in a polypeptide, because protein synthesis in cells occurs at site where DNA is absent, right? So where does the protein synthesis or the translation occur? It occurs in the cytoplasm. Where is the DNA? It is inside the nucleus, right? Example, protein synthesis happens in cytoplasm, whereas DNA is in nucleus, which is separated by a membrane, right? So there must be another information conveying molecule that obtains specific genetic information from DNA, and it moves to the cytoplasm to act as a template for protein synthesis, right? The most likely candidate for this looked like RNA, right? So RNA seemed like a likely candidate for such a messenger molecule. Why? Because what is RNA? It is another class of nucleic acid resembling DNA to some extent, right? We know that the only major difference is that there is a oxygen present in C2 position, and also along with that, instead of thymine, we have uracil in case of RNA, right? These are the two major differences. Then it was possible to imagine DNA serving as a template for its synthesis. Uh, it is abundant in the cytoplasm. RNA is an unbranched polymer of ribonucleotides, that is AUGC are present, and it is linked by 3' to 5' phosphodiester bonds. Also, it contains ribose sugar at hydroxyl group C2. It also lacked thymine instead of uracil, which is similar to T, except that it lacks methyl group at the 5' position in the pyrimidine ring, which is replaced by a hydrogen. In the cell, RNA is usually present as a single-stranded molecule, and double-stranded helices can be formed intramolecularly by pairing of bases in different regions of the same RNA molecule. When this particular RNA was discovered as the messenger that is actually conveying information from DNA to protein, the central dogma was proposed, right? In the central dogma, it was said that DNA can replicate itself and it can form more DNA, or it can form RNA through transcription. This RNA can form a protein or instruct formation of proteins through translation, right? And it was said that this is a unidirectional process, right? So only DNA can form itself, but others is the other part is a straight line, that is, it is unidirectional. It cannot happen in the opposite direction. That is, only DNA can form RNA, only RNA can form protein, right? Uh, and After this, Francis Crick also hypothesized another, mechanism of how protein would be formed, how, these codes will be, or how information will be recognized from RNA, and how protein will be translated, right? And in this is known as the adapter hypothesis. And the adapter portion in this is the transfer RNA or the tRNA. We will read about it. So it seemed unlikely that the RNA template would be able to distinguish between very similar amino acids like glutamine or valine. But prior to incorporation into proteins, amino acids must first attach to specific adapters. These adapters would then specifically bind bases on RNA templates, which could be short RNAs. Then Paul Zamaknik and Malon Hogland further added that amino acids are first attached to tRNA molecules prior to incorporation into polypeptide chains. Also, each tRNA has a short sequence of adjacent bases, which are known as anticodon, that can bind specifically to complementary groups of adjacent bases, which are codons, on the mRNA template that could specify the amino acid sequence. The set of three bases specify amino acid that should be added, which is the triplet code and is referred to as the codon. Now, with reference to this, we have also discussed the Vogel hypothesis in the practice session. I would refer that to you again. Also, a full set of codons corresponding to amino acids, the genetic code, was determined by the efforts of several scientists, such as Marshall Nirenberg, Heinrich Matthey, and Hargobind Khurana. Uh, After this, there came another major discovery wherein this scientist, that is Howard Temin, basically proposed that the provirus is a DNA copy of viral RNA genome. Right, that sometimes RNA can also form DNA. Right, now this was something that was going against central dogma. In 1970, Temin and Mizutani discovered a viral enzyme, that is reverse transcriptase, that is synthesizes DNA from RNA. This was an unambiguous evidence in support of pro-virus hypothesis that a key tenet of the central dogma, that is unidirectional flow of information from DNA to RNA, could be reversed. Right. So this basically forms a DNA from an RNA, right? And then that DNA is actually going and attacking the host genome. So this is what a retrovirus was. And it basically caused revisions to occur in the central dogma, wherein now it was seen that translation could, even mRNA could replicate like DNA was replicating, and it could also go in the opposite direction, right? Apart from that, there are other revisions that have been done in central dogma that we have not talked about over here. One of which is that RNA can itself also, some kinds of RNA can act as a protein, right? So, for example, enzymes are majorly proteins, right? But some RNAs can also act as enzymes. These are riboenzymes, right? So that is another revision in the central dogma. Then comes the transcriptional initiation in eukaryotes. So over here you can see that there is a complex that is present, and it has to be assembled in this particular format. The transcription factor 2D, the transcription factor binding protein to V2A, the polymerase with its multiple units have to be present in a complex form, right? So that there will be initiation of the transcription that will occur, right? And these come and bind to a specific region which is known as starter box, and there are some, adapter molecules that are present over here, which basically turn the DNA or fold the DNA in such a way that the enhancer comes uh, close by to the site where the transcription actually has to initiate, so that this enhancer will signal the complete binding of this functional unit and then initiation of transcription. Okay. Then the transcription machinery in eukaryotes, like we talked about, it had these. That was a transcriptional machinery. This is a translational machinery. So after transcription, what will we get? We will get an mRNA, right? Which is complementary to the coding strand of, to the non-coding strand of DNA, right? So, after transcription, we will get an mRNA, and after that, it will go some modifications. There will be 5' capping and 3' tail that will be attached, right? So that it can now go to the cytoplasm where the translation will actually occur, right? There is some translational machinery that are present in eukaryotes. Now, they are the tRNAs, mature tRNAs that have to be present, the ribosomes that have to be present, right? So these ribosomes they will basically go and uh, they will assemble the amino acids that are being brought by these tRNAs. After these tRNAs have read the code on the mRNA, right? They will go and they will take some whatever is the next amino acid to this ribosome, and ribosome will attach these amino acids together. Right? That is how translation will be done, and this is the translational machinery of eukaryotes. Then there are certain steps that are present in protein translation. They include these three major sites in the RNA, right? So one is the amino acid site, one is the peptide bond formation site, the other is the exit site that is present, right? So the first amino acid that will come, it will basically sit in the amino acid site, right? After that, it will move to the peptidyl site, and there will be initiation of protein translation, right? So, oh, it will there will be initiation of protein translation. After that, there will be elongation, right? So a new protein will come and sit over here. And this one will go to the exit side. Exit side will remove it, and they will keep shifting, and the amino acids will keep getting attached, right? So the tRNA is now free. It will go and read another sequence, and the amino acids are all fixed over here, right? And then whenever a stop codon or a stop signal is received, there will be no further recruitment of any tRNA over here, and there will be termination of the signal, and the polypeptide that is made will be set free. The translation in prokaryotes, The prokaryotes don't have nuclear membrane compartmentalization, so the transcription and translation machineries are basically present in the same location, right? So ribosomes start building proteins as the mRNA is being transcribed. So since there is no division over here, right, it is not like it has to come out of the nucleus or the nuclear envelope and then it will enter cytoplasm, then the ribosomes will access it. No, in prokaryotes, that division is not there, right? So as the mRNA is being synthesized, simultaneously tRNA will come, it will read, and the protein synthesis will also start. The ribosome will also sit and it will also start a protein synthesis. So that occurs. So, transcription elongation factors link the RNA polymerase to the ribosome, holding it close but not too close. The RNA polymerase, a ribosome, and associated molecules together form expressosome or a TTC, that is transcription translation complex, right? So it will occur simultaneously over here. Then, after briefly having talked about how the transcription translation works in case of prokaryotes, we need to understand how cells basically replicate or how they reproduce, right? So there are duplication of all the constituents of the cell. Then after they are duplicated, they have to be segregated properly, right? Uh, okay, failed. What? So after everything has been duplicated, it needs to be put into different compartments, right? It needs to be segregated. After it is segregated, there will be a division into the daughter cells. So after it is seen that, okay, everything that was duplicated, this cell has gotten one copy and this has gotten another copy. Now we can actually physically divide these two cells and they will become two different daughter cells. Now, this is the basic of cell cycle that I think all of us now know, even if we were initially unaware about it. So now we know that the cell cycle has these four major phases. One is the gap one phase, then the synthesis phase, then gap two, and then mitosis. And if a cell is exiting the cell cycle and it is not a mitotically active cell, then it will go into this G0 phase or the quiescent state, right? In the G1 phase, which is the first growth phase, which is prior to DNA replication, it is highly regulated and usually it is the longest phase, right? After this comes the DNA synthesis phase where replication of DNA occurs. There are new histones and chromosomal proteins also that are formed. Then comes the second gap phase where growth phase prior to active separation of chromosomes occurs. Then the synthesis of M phase proteins also takes place in this phase only. Then comes mitosis. This is a short phase, but it is very, very active. And the separation of chromosomes and cells, that is cytokinesis and karyokinesis, occurs over here. Uh, then we know that there is a cell cycle. Now, we also know that this cell cycle is regulated, right? There is something that is making this cell cycle go in this order only, right? And what is this? How do we know that it is being regulated? There were certain evidences for it, right? So evidence one that we actually got was from this paper that was published in 1917 in 1970 in Nature by Johnson and Rao, and it basically said that first, if we see that G1 nucleus enters the S phase immediately, right? And S nucleus continues DNA replication. That is what happens over here, right? So if there is an S phase cell that is present and G1 cell phase that is present, it will be seen that the G1 will also enter the S phase and both of them are now in the S phase. If there is a S phase cell that is present and G2 phase cell that is present, it will be seen that G2 cell stays in G2 and S continues to replicate, right? G2 will also not convert into S phase. And the S phase, since the division is still going on, it will also not readily convert into G2, right? Until this division is done. Also, when it is seen that G1 is added to G2, the G2 stays in G2 while the G1 enters S as per its own timetable. Because over here, the the regulatory factors that would have pushed it to enter S phase in the first case, because the other cell was already in S phase and it would already have those factors, are not present in case of G2, right? So it basically told us that S phase cell induces premature DNA synthesis in G1. That is replication promoter factor. Replication promoting factor is probably present in S. And S phase cell cannot induce DNA synthesis in G2 because probably re-replication is blocked in G2, right? It cannot maybe enter the previous phase. That is what we saw in this study. Then we come to evidence for regulation. That is the second evidence that came from this paper in 1971 by Massoe and Marquardt. Right. What they did first was that xenopus egg was taken and its maturation and activation was studied. Right. So once its egg was taken, it was an oocyte which was arrested in G2 phase of meiosis 1. There was a hormonal trigger that was given and it entered the M phase of meiosis 1. Then in after that it was arrested in M phase of meiosis 2. Right? After that, it was activated such that there is fertilization by the presence of a sperm near this oocyte, and after fertilization the cell cycle phases were completed and cleavage actually took place, right? And it was seen in the same paper, this factor that is known as maturation promoting factor, or MPF, was defined, or it was actually discovered. What it does was that the cytoplasm from M phase oocyte was actually taken and it was given to another oocyte which was in interphase, and it was seen that the oocyte remains in the G2 phase, right? Even if this M phase cell is actually oh, sorry. So in case of G2 phase, if M phase cytoplasm is actually injected, it is driven into M phase. But in that particular oocyte, if cytoplasm from interphase is injected, it does not have any factors that promote mitosis, so it will remain in G2 phase. So it basically it was concluded that a cytoplasmic regulator, that is MPF, or maturation promoting factor, will control entry into mitosis, right? Later it was studied in more detail, we will come to that again. And it was identified that there was a master regulator of the cell cycle. What was this master regulator? It was a complex of cyclins and CDKs. Right? CDKs are cyclin dependent kinases. So cyclin was one protein and the kinases are dependent on it. These were basically master regulators of the cell cycle. And it was basically discovered by Leland Hartwell, Paul Nurse, and Tim Hunt. Also, another important point that was discussed during this lecture by MAP was that in G1 phase, which is the longest phase actually, there is a point which in Saccharomyces cerevisiae or yeast is known as the start site, and in higher eukaryotes or us it is known as the restriction point. And this point comes towards almost towards the end of the G1 phase, and once this point is actually reached or it is crossed, the cell is determined to undergo a cell division. Right? Before this, the cell can still enter the quiescent phase and it might not undergo a cell division. But once this point is crossed, it is one of the major checkpoints and it will determine that, okay, the cell is definitely going to divide. Then during this lecture also we discussed about yeast life cycle, which again we have discussed very recently in week 8 lectures, right? So we know that yeast has two kinds of sexes that are present. One is an alpha, the other is A, right? So they can undergo sexual reproduction or asexual reproduction. In asexual, they will just keep budding from one sex or one mating type only. In sexual reproduction, however, two different mating types will come together. There will be conjugation of these two different mating types that will occur, and a diploid yeast will be formed, right? And these diploid yeast will also undergo a spore formation, and it usually occurs when the conditions are unfavorable for these yeast to survive. Again, why it is so, we have already discussed in the practice session. Then we come to mutagenesis and screening. So yeast, why we have described its life cycle over here is because yeast is one of the organisms that is readily growing, and it is very easy to, since it is readily dividing, it is easy and it is a good model to actually study cell division or cell cycle in. Okay. So, how was this study done in yeast? So, what was done was that mutants from yeast were taken, and it was seen that what is the molecular level change that is there in this particular mutant, right? So, how was the screening done? The yeast cells were mutagenized using mutagens such as EMS. Then mutagenized both the alpha and the A cells, and they screened and selected for desired phenotype, which is a primary screening. Then in the secondary, they screen for desired phenotype again, and the cross mutants to assign complementation groups. Then they identify gene by complementation from a gene expression library. So for CDC mutants, the primary phenotype was temperature sensitivity, which was conditional mutant, and the secondary was microscopy for cell cycle arrest phenotypes. In the Saccharomyces cerevisiae, it was seen that although the wild type looked like this, it could be in different phases of cell division and budding could be happening. If there were terminal phenotypes that were present in the CDC mutants. If there was a CDC1 mutant, it looked like this, right? Uh, uh, the budding was restricted in this case. CDC 28, again, that happens. In CDC 14, the budding actually was happening, or the cell cycle division was trying to occur, but it was not being able to complete this division. And in CDC 15, there was another kind of phenotype that was seen, right? Similar, okay. And they were divided into functional categories, such as the controls responsible for progress of cell cycle, processes monitored by checkpoints, signaling pathways that cause arrest in response to external signals, and morphogenetic steps of the cell cycle. In the CDC 28 mutant, it involved a major cell cycle control called START. A G1 arrested at CDC 28 cells block progress at start and can still conjugate when released from G1 arrest at the restrictive temperature. So it means that if it is at a certain temperature or a restrictive temperature, it will uh, be restricted at the start site, but it can surpass or bypass the start site when optimal temperature is given. Then the CDC 28 do not initiate DNA synthesis, right? Why? Because CDC 28 encodes a CDK, that is cyclin dependent kinase. Then there is another species of yeast that is present, that is Saccharomyces pompe, not cerevisiae. If what happens in this is, instead of budding, there is fragmentation that takes place. And it was seen that in the wild type, the fragmentation was normal, but in the CDC 2 deficient kind, or V1 negative, or OPCDC 21 positive, the separation couldn't take place. There was no, the the starting of the cell was very small. It was not as a normal wild type cell, and then again it could not be separated even after the division had occurred. On the contrary, in CDC2 negative, CDC 27 negative, OPV1 positive, and CDC 13 negative, it was seen that although the division has occurred over here, so there is DNA replication probably that has occurred, but the division of cell has not taken place. So CDC2 basically encoded a 34 kilodalton phosphoprotein. P43 levels and phosphorylation do not change during mitosis, and P43 levels dephosphorylated on entry to stationary phase. P34, not 43, P34 phosphorylation correlates with high mitotic index, that is septated cells. And P34 has kinase activity, which is thermolabile in CDC2TS mutant. These were the mutants that were found. After this, Tim Hunt, it was also discovered in sea urchins and clams that cyclins are present. Now, what is this cyclin? It is basically, or it is named basically because it is going up and down. So it is in a cyclic manner. It is going up and down in response to whether the cell is in interphase or it is in mitosis. Thus, the name cyclin was given. And the first cyclin to be discovered by them was the MPF, or maturation promoting factor, right? Which basically went up during the mitosis phase and then it declined again in the interphase. When we talk about MPF, it induces immature oocytes to become an egg. We have recently just seen that experiment. Its biochemical purification revealed that it had two subunits. It consists of cyclin-dependent protein kinase and cyclin B, and CDK was always present. Cyclin levels sometimes fluctuated, right? So it means that CDKs might be present, but for them to perform some activity, cyclins are present, and cyclins are not always present. Their levels are fluctuating. MP, uh, MPF activity is transient, so it is probably a cyclin. A cyclin is a regulatory subunit and it activates CDK. CDK induces mitosis by phosphorylating specific target proteins on the serine and threonine. What are CDKs? They are cyclin-dependent kinases. Their activity is regulated by cyclin association, which activates CDK2. The inhibitory phosphorylation on threonine 14 and tyrosine 153, and activating phosphorylation on threonine 1614. Also, there are some inhibitors of these CDKs, that is CKIs, which when they bind to CDK, it inhibits the CDK mediated phosphorylation. In the S. pombe, which was the second kind of Saccharomyces or yeast that we saw, in the CDC2 mutant, the cyclin-dependent kinase, CDC2 is a cyclin-dependent kinase, which is equivalent to CDC28 of Saccharomyces cerevisiae. And CDC13 is actually the cyclin. So if CDC13 is not present, even if the CDC2 or CDC28 is present, it is not going to work in absence of CDC13. Also, V1 is an inhibitory kinase and CDC25 is a phosphatase which activates the CDC213 complex, which is what is shown in this representation also. Then how do cyclin levels cycle? How does it happen? Because probably when the level is dipping, it is being removed or it is being degraded or it is being pushed out of that particular cell, right? So the major player in their levels cycling is ubiquitin. Ubiquitin is another 76 amino acid polypeptide. It is covalently conjugated to substrate lysine residues by E1, E2, and E3 enzymes. The ubiquitinated proteins are degraded by the 26th proteasome. There are two E3 complexes that are involved in cell cycle regulation. One is the SCF and the other is the APC. Right? SCF is a modular E3 in which is associated with E2 CDC34, and it is the substrates or a substrate of G1 cyclins and certain inhibitory complexes, I kappa beta, E2F, etc. Then there is APC, which is an E3 enzyme with 12 subunits. And the destruction of APC requires presence of 9 amino acid motif called the destruction box. It is a substrate of M cyclin and PDS1. Then how does the phosphorylation affect the CDK1 activity? When a phosphate is present, it can cause steric blockade of the peptide substrate binding, and this binding can create an environment that favors a non-productive conformation of terminal group of ATP. This phosphorylation inhibits, but it retains trace amount of activity. the speculation that how this might be occurring is that additional phosphorylation at threonine 14 may further block the peptide binding site and may also prevent the binding or appropriate conformation of the configuration of the ATP. So it basically is speculated that it is going and physically blocking the site of phosphorylation, right? Or physically blocking that site. There was another interesting prediction or another interesting points that was discussed by these authors in their paper that despite this, the phosphorylation of threonine 160 leads to conformational change that embeds the phosphate group in a network of stable interactions, which is required for structural integrity. The tyrosine 15 phosphate moiety remains solvent exposed and is readily acceptable to both V1 or MIT1 kinases and CDC25 phosphatases. Thus, increased CDK tyrosine phosphorylation provides the cell with rapid and potentially reversible mechanism for regulating CDK activity in response to a checkpoint activation. Then there is T160 activation that is embedded in this phosphorylation may be harder to remove once it was made, right? So anyway, I'm just skipping to the next slide that what could be the mechanism of inhibition of CDK by CKI is that it is physically going and it is blocking this active site, right? For example, this one kind of CKI that is INK4, it could cause active site loop to move and cause inactivation and impacting interaction with cyclin. In P27 KIP1, what would happen that binding invades into the active site and blocking the ATP binding site. So that is essentially what they are doing. Then this is a list of cyclins and CDKs of the vertebrates and their counterparts in Saccharomyces cerevisiae, right? So you can go through this list. Also, it is seen that CDK activation will drive the cell cycle forward, right? How will it do it? That on activation of CDK, it phosphorylates several of its target proteins. The phosphorylation will change the properties of these targets and it may inactivate or activate their functions, or it will affect their interactions with other molecules. These changes may bring about events required for cell cycle progression. So, for example, what could happen that lamin filament, which is present on the inner side of the nuclear envelope, it on the in the presence of CDK1 cyclin B, it will dissociate these lamin dimers and it will slowly dissociate the nuclear membrane. And the dissolution of nuclear envelope occurs that we know when the cell is undergoing mitosis, right? Then there are certain cycle checkpoints that are present, like we have already discussed that we know that there is some amount of regulation in the cell cycle, and it is probably due to some kind of control or checkpoint. So checkpoint controls are signaling pathways that act to delay cell cycle progression when perturbations delay completion of certain cell cycle events. So what does that mean? That if there is some thing that has not been done correctly or there is some hindrance or something is not completed yet, it will not let the cell progress to another stage. That is what this checkpoint control will do, right? So checkpoint pathways involve detection of errors or problems in the cellular process, such as DNA damage or spindle attachment. It will also involve transmission of the signal and effector that inhibits the cell cycle progression. Also, checkpoints slow down or arrest the cell cycle to allow the repair of damage before it proceeds. The checkpoint mechanisms are critical for fidelity of ongoing cell division, and the mutations in some checkpoint genes have been associated with cancer predisposition and progression. A cell that is unable to fix the damage may undergo apoptosis. Then we study about cell cycle control. How does it happen that the events must be turned on at an appropriate time? The requisite time has to be available for completion of the process. Events occur in a particular order. There must be a mechanism to ensure this. Each major event must be triggered only once per cycle, right? So if a cell cycle division is already on the go, it will not, the cell control checkpoints will not allow another division to initiate, right? Then binary on-off switches that trigger events in complete reversible fashion, and backup mechanisms should be present to ensure robustness. Even if one fails, the other can compensate for it, and adaptation to respond to specific environmental conditions or to perform specialized functions uh, would probably be present in cell cycle control, right? What are the key regulators of cell cycle control? One that we have read is the CDK cycling complex. They will phosphorylate specific targets to trigger the cell cycle events. Also, they will drive the cell cycle forward when they are active. So it is a positive regulator. The negative regulator is the checkpoints, which will prevent the entry to the next phase until the events in the preceding phase have been completed. Also, they will inhibit the progression of cell cycle when they are active. Then there are some key concepts related to checkpoints that were given by Hartwell. He said that the cell cycle should be considered as temporarily organized sequence of events linked together in orderly fashion. The later the events dependent on successful completion of earlier events will occur. Two types of dependencies occur. One is direct coupling and the other is linking signaling control, which is the checkpoints. Then there are certain events which could be rate limiting. Uh, then these checkpoints, as defined by Hartwell and Wiener, are checkpoints controls the signaling pathways that act to delay cell cycle progression when perturbations delay completion of cell cycle events. That is what we have read earlier also. Then checkpoint dependencies can be established in either of the two ways. One model is the substrate product model and the other is the checkpoint control model. What was the first model that was proposed? It used to define a mechanistic linkage. For example, completion of one event produces substrate necessary for execution of the second event, right? But if this kind of control occurred, it will be very hard to uncouple, right? So because it has to after the cell cycle has been completed, it has to undergo another round of cell cycle. So it will be easy to follow up in the first place. In the first cell cycle it will be easy to regulate it, but after that it will be very hard to uncouple. The other was a checkpoint control model, which said that it is mechanistically unlinked. A regulatory pathway ensures that later events does not begin earlier until the earlier one is completed. And this model seemed to be easier to uncouple. So there was a prediction that a null allele in checkpoint genes should result in loss of dependency, right? So if it means that if there's a loss of function of checkpoint genes, it will not, the cell cycle will not be dependent at all, right? So that means that the checkpoints are inhibitory pathways. So there will be no inhibition of cell cycle whatsoever, even if there's DNA damage. Incorrect DNA will only be undergo cell cycle in that case. So, what are the functional requirements for checkpoint response? One is to monitor or sensor. So, it detects detects progress or completion of one cell cycle event. The other is signal transducer or relaying information. The signaling components to relay signal to effector, and the third is effector or target, that is regulator of initiation of subsequent events. Right. Then there are certain events that are linked in the dependent pathways. And the question was that whether they are mechanistic or they are non-mechanistic or checkpoint control mechanisms. And through experiments and through, multiple reasonings, it has come up that these are in fact checkpoint control mechanisms which are not mechanistic, right? So that they can easily be uncoupled later. So the first kind of checkpoint that we can discuss, the first we have already discussed actually, it is the start site or the restriction site. That could also be considered as a checkpoint, right? The first major checkpoint after that is the DNA damage checkpoint. What it does is that if there is DNA damage, it will basically stimulate or it will activate RAD9P, RAD17P, RAD24P, and MEC3P. And these will basically not let the uh, the cell cross the start phase or allow DNA replication to occur further, or let the metaphase to anaphase transition to actually occur, right? So these operate in interphase. There is G1, S, and G2 phases. Sense damaged DNA and prevent they sense damaged DNA and prevent progression till the DNA has been repaired. The G1 checkpoint allow the time for repair to happen prior to entry into the S phase, and the S phase checkpoints monitor damage during the DNA replication. The G2 checkpoint senses damaged DNA or incomplete replication in G2 and prevents initiation of M phase until replication is complete and any damage has been repaired. Right? So these DNA damage checkpoints will induce cell cycle arrest if there is unreplicated or single standard DNA that is present through ATR and through phosphorylating check 1, and if there's a double stranded break that is present through ATM and phosphorylating check 2, right? Both of them will not let the binding of CDC 25 to occur to the CDK, so that there will be no further, uh, beginning of the cell cycle events. Now, what is the role of P53 in the cell cycle arrest when there is some kind of DNA damage, right? The P53, it basically no, it will basically be silenced by the presence of this MDM2, right? When there is some kind of damage that is present, this silence protein MDM2 will be ubiquitin annihilated and it will be degraded by the ubiquitin proteasomal mechanism, and the P53 will be phosphorylated. Now, this will basically act P53, which will go and it will bind this double-stranded break where the DNA where the double-stranded break has occurred, right? Once it will bind, it will basically stimulate the activity of P21 mRNA and it will inhibit CDK2, cyclin E or cyclin A complex. And thus the cell cycle progression will be inhibited, right? And this will sit over here until this DNA is repaired. So DNA replication checkpoint also monitors completion of the S phase. We over here is another experiment wherein there is a normal S phase when there is an untreated cell, that there is synthesis G2 and a normal mitotic phase. Even when caffeine is given, the stages are more or less similar, and they that is how it progresses. There are two daughter cells that are still present. But when hydroxyurea or HU is given, the S phase keeps on prolonging. And since there is delay in S phase, there is no mitosis that is occurring and only one cell is actually present, right? But when HU is actually given with caffeine, then this S phase, which was prolonging in the presence of caffeine, it will even if the DNA is not correct, right? Even if it should not undergo mitosis, it will lead to for mitosis to occur. And because this was incorrect DNA or it was unnatural kind of DNA, it is known as suicidal mitosis, and it will occur, and the kind of genetic material that is present in the two daughter cells uh, will make both these cells non-viable, right? So HU or hydroxyurea is an inhibitor of ribonucleotide reductase, and it causes delay in S phase progression due to depletion of the DNTPase, right? So HU causes S phase arrest, and cells are arrested with a short spindle. So the screen over here was that HU sensitive mutants that show elongated spindles in presence of hydroxyurea are likely to be defective in mechanism that arrests in response to replication stress, right? Even if HU is given, but there is normal spindle or elongated spindle that is present in the cells, then it means that they cannot sense this HU, right? So this mutant was basically known as MRC1 mutant. It is HU sensitive. It elongates spindles even in presence of HU, and it fails to phosphorylate RAD53. That is how it elongates spindles. In response to HU or MMS treatment, unlike wild type cells. So MRC1's phosphorylation depends on ATR homologue MEC1 and SQ or TQ cluster domain target phosphorylation sites for kinases of ATR class. These mutants show compromised viability in presence of replication stress. So it means that although it is able to rescue the phenotype, but it will have compromised viability because there was some loss of DNTPs that was definitely there, and it was not a normal replication process that went on, right? And also, this variant fails to activate RAD53 in response to replication stress. If we talk more about this particular variant, MRC1, the mediator of replication checkpoint, we can see that mutants of this variant are replication checkpoint effective and fail to activate RAD53 in response to replication stress, like we just saw. The checkpoint and replication phenotypes of MRC1 are separable, and replication initiation is required for MRC1's chromatin association. MRC1 moves with replication fork. The checkpoint activity is not required for localization to replication forks, and MEC1 is recruited to sites of DNA replication interface. MRC1 phosphorylation is in response to DNA replication stress is required for MEC1 accumulation at the stalked fork. Then there were this other class of proteins that was MAD proteins, which had been shown to regulate mitosis. So the full form of this MAD protein is mitotic arrest deficient mutant. So they could not arrest the cells to not undergo mitosis, even if there was a defect that was present, right? So this Benomyl sensitive mutants in which completion of mitosis is not delayed in presence of benomyl. These are mutants that die due to premature exit in presence of benomyl. Then they divide more rapidly than wild type to form 20 to 50 cell microcolonies and increase frequency of chromosome loss, right? Although they are forming multiple cells, they are undergoing cell division, they are not intact or viable cells. There is chromosome loss in these cells, right? There are these various types of MAD proteins that are present. So I will just finish this particular topic, right? And I will just share the material with you guys after that. Then there are another kinds of mutants that were found, which were the BUB mutants. These are the budding uninhibited by benzimidazole. So it is very similar to the MAD mutants that were present, right? Over they were also active mitotically despite there being defects that are present. This is a similar case. And compounds such as benomyl and ocodazole cause disruption of microtubule structure. If this disruption occurs, there is a arrest as large budded cells with a single 2N nucleus. Then cytokinesis and G1 of next cell cycle, that is new bud emergence, DNA replication, SPB duplication are inhibited. But in case of BUB mutants, new bud emergence and DNA replication occur in presence of benomyl, although there is no low survival in nocodazole containing media. So this is the BUB mutant, right? So the buds keep on emerging, although they are not separating from one another, but keep on emerging. Then comes the spindle assembly checkpoint. I will quickly go through this one also. I'll just finish checkpoints, right? So in spindle assembly checkpoint, what happens during the mitosis when it is occurring, when it has to switch from metaphase to anaphase, there has to be a checkpoint wherein this check occurs that whether the spindle has attached correctly to both the sister chromatids, right? So that if it has attached properly, it can be pulled to the opposite poles. But if this attachment has not occurred, or if it is not correctly occurred, then there might be pulling of both the sister chromatids to one pole, or it cannot be pulled to the opposite pole, something like that. Some defect might occur, right? Like this. So for that, there is a spindle assembly checkpoint, which basically monitors this attachment of spindle to this sister chromatids, right? So that is present. That is the if there is a incorrect attachment, it will basically inhibit the anaphase promoting complex, and thus this cannot progress to anaphase, right, until this attachment is proper. So these are the various checkpoints that were there, right? There are multiple DNA damage checkpoints present in G1, S, and G2 phase. Once the cell actually enters mitosis, then there is spindle assembly checkpoint that is present between a metaphase to anaphase transition. Uh, then is the ubiquitin proteasome system, but I will just take your leave over here. Right, it is a lot to discuss for 2 hours. Um, as I said in the beginning of today's lecture, what I'll do instead is, okay, I'm still sharing my screen. No, yeah, okay, I'm not sharing my screen, I think. So, yeah, as I told you in the beginning of the class, what I'll try to do is I did prepare the material for 5 or 6 weeks, I think already. So I will add the material for all 8 weeks into one particular PowerPoint presentation or PDF. Most of the slides will be there, right? I have taken snapshots of a lot of slides and it will be there. So for those people who were actually complaining about not getting the PDF of this course, at least that complaint will not be there. So if you are reading for your exam, uh, uh, in the last moment, at least you can go through that PDF only, if not all lectures. So that I'll do. With that, I will just end today's session. It is a lot of course to be summarized within 2 hours. I think I will need 3 such classes, but that is not an option. So that is it from my end for this particular course. I will share the material with you guys. Apart from that, thank you so much for being with me through this course. And I hope you have learned a lot from ma'am's lectures, from the practice sessions, from all the TAs, right? And that is it. All the best for your exams, and see you. Good night.
Thank you, ma'am.